Hello, everyone.
Hi.
Good to see you. If you wouldn't mind, go ahead and type your first name or your nickname into chat for me. Thank you. Thank you very much. That's great. All right. Okay, so today we're going to be talking about the topic of fermentation. We're going to continue talking about carbohydrate a little bit. Before we get started looking at the lab material, I'm just going to pull up our schedule so we can take a look at what's happening and what's coming up for us. So this is just the schedule that you can see at the end of the syllabus. You can see we're right here at week 8. This week in lecture, you've got 2 different topics about microbial metabolism. Those are both the topic of enzymes and energy, and also the topic of carbohydrate catabolism in terms of cellular respiration and oxidative phosphorylation, and this process called fermentation. So a lot of metabolism this week. As always, if you have any questions while you're watching lecture material, be sure to jot them down and bring them to lab, or you can send me a message through Canvas and we can talk about it together. It's a difficult topic, especially if you don't have a lot of background talking about these processes. It may be a long time since you talked about things like cellular respiration. So, all right. So does anybody have any questions about anything? First of all, anything that you want to ask about or talk about, feel free to type it into the chat. I've been grading exams, so remember that when you get your exams returned to you, lecture exams and lab practical exams, you'll always want to, when you pull the exam back up, there's a place where you can click that says view instructor comments. Remember, you always want to do that to see if I've left you any comments about what you might have missed. It's a good opportunity to figure out why you might have gotten something wrong. And of course, if you ever miss something on an exam or a lab practical and you don't understand why, message me and we'll talk about it. Okay. All right. So when we talk about carbohydrate catabolism, again, that's the branch of metabolism where carbohydrates are being broken down by microbes. Fermentation is one of the big processes that we talk about because many microbes use fermentation. They metabolize carbohydrates, some of them, in multiple ways. Some of them do cellular respiration aerobically or anaerobically, and many microbes are also able to ferment. And what makes them particularly interesting and diverse is that some microbes can ferment several different sugars. One of the ways that we test for fermentation is by testing their ability to ferment glucose, but we also test their ability to ferment other sugars, usually disaccharides, things like lactose or fructose or sucrose, these very common sugars that they might encounter in their environment. Remember that when we say that a bacterium can ferment something like lactose, what we're really saying is that they have the enzymes necessary to take the lactose molecule and break it in half into the two simple sugars that are inside that disaccharide. If you don't remember, lactose is actually made from two sugars called glucose and galactose. And they come together with a covalent bond and they make lactose or milk sugar. So some microbes have the ability to break that bond in lactose and set free the glucose and the galactose. And then they can take that glucose and they can ferment it. And when we talk about fermentation, the very first part of fermentation involves the same process that we see at the very first part of cellular respiration. And that's a process called glycolysis. It's actually a series of processes. It's a 10-step reaction. It requires 10 different enzymes. But the glucose molecule, as it goes through glycolysis, is converted into a molecule called pyruvate. And of course, during cellular respiration, pyruvate is going to continue along towards pyruvate processing and the Krebs cycle. But in fermentation, the pyruvate takes a different path. So both cellular respiration and fermentation begin with glycolysis, with glucose being converted to pyruvate. The different types of fermentation then come from the fact that the pyruvate can be converted into lots of different products. Pyruvate can be converted into things like acids and alcohols and gases. Those are the three big products of fermentation: acids, alcohols, and gases. So different types of fermentation are going to produce different combinations of those products. No problem, Chloe. Glad to have you. For example, you and I, we're capable of fermenting, aren't we? We're capable of lactic acid fermentation. Now, we normally, in our cells, we normally do aerobic cellular respiration. So we use oxygen as our final electron acceptor during cellular respiration. As glucose gets oxidized, that just means broken apart, essentially. At the end of all of the reactions, something has to get reduced because oxidation and reduction go hand in hand. If something gets oxidized, something else has to be reduced. In other words, if something's going to lose electrons, that's oxidation. Something else has to gain those electrons. That's reduction. And at the very, very end of the electron transport chain in our mitochondria, all of the electrons that get removed from glucose, as the glucose is broken up, all of those electrons have to be accepted by something, and that something is oxygen. That's how it works for you and I. But there are times when we can't get enough oxygen, right? There are times when we just can't get enough oxygen to meet our needs. We are big, complex organisms. We have millions and billions of cells in us. And sometimes we do things that just push our ability to breathe. Think about, the last time you were doing an exercise that was very, very difficult for you. Maybe you were running and you got very, very tired and you were just gasping for air. The reason you're breathing so fast is you're trying to get enough oxygen in to make enough ATP so you can keep running. And sometimes we just can't. Sometimes we simply cannot get enough. So our cells will begin to ferment. And the type of fermentation that our cells are capable of is lactic acid fermentation. So the glucose goes through glycolysis and becomes pyruvate. And then the pyruvate is sent into the lactic acid pathway. Now, we're not designed to ferment for very long. It's not going to keep us alive all by itself. If you can't get enough oxygen, you're not going to survive. But in humans, fermentation is sort of a secondary pathway that our cells can draw on at times when they just can't do respiration fast enough because we can't get enough oxygen. Microbes are different. There are many microbes that only ferment. It's the only pathway they use. They just don't have as high a need for ATP as you and I do. They're just not the same kind of organism as we are. So there are some microbes who use fermentation exclusively, but there are lots of other microbes that use fermentation as one of their pathways to make ATP. There are lots of microbes that can do respiration and fermentation. There are microbes that can do aerobic respiration, anaerobic respiration, and fermentation, and they can switch pretty well between those pathways. So they're very, very diverse, metabolically speaking. So when it comes to fermentation, we test microbes to see what they can ferment, which sugars, which simple sugars are they able to ferment, and what products are they making when they ferment. Those are characteristics that individual microbes have, and they can help us identify those microbes. And we can use different tests in order to find out what abilities they have in terms of fermentation. And that's what we'll talk about today. So I'm going to go ahead and pull up our slides. That's what you should be seeing on your screen now is the title slide for the fermentation exercise. We need to define this process. We need to have a definition for what fermentation is. Fermentation is an anaerobic process that converts the product of glycolysis, which is pyruvate, into some combination of acid, alcohol, and or gas. Now, it's an anaerobic process. That's important. Even an aerobic microbe, even a microbe that requires oxygen, like you and I do, when they do glycolysis doesn't require oxygen. It happens without oxygen. It doesn't need oxygen, in other words, to happen. So the first part of respiration and the first part of fermentation, this is an anaerobic process, this process of glycolysis. Now, the question is, why would a microbe ferment? Why would a microbe evolve in order to use fermentation as their metabolic pathway or one of their metabolic pathways? So there are three reasons here. The first reason is to produce energy, again, in the form of ATP, when cellular respiration is just not possible for them. So that's a situation that would be very similar to what you find in a human being. There are microbes who normally do cellular respiration, either aerobic or anaerobic, but they find themselves in a situation where they can't get enough of their final electron acceptor, whatever that happens to be, whether it's oxygen or one of the anaerobic ones like nitrate. If they can't get enough of their final electron acceptor, they can't do cellular respiration. It won't happen. So they turned to fermentation as a secondary pathway. so they can still keep making ATP when they're not able to do respiration. That's the first reason. Other microbes are going to use fermentation to produce, again, ATP as an equal alternative to respiration. In other words, some microbes don't use fermentation only as a salvage pathway because they can't do respiration. Some microbes do both pathways equally well. So it just depends on what environment they're in, whether they're going to use respiration or fermentation. And then there's a third class of microbe. Some microbes are pure fermenters. In other words, they only use fermentation to make their ATP. So we see all different kinds. But the idea is the same here. The reason that a microbe ferments is to make ATP. They need ATP. All living things need ATP to survive. We just make it differently. So we got to keep those three pathways separate in our head now. We have to be able to keep respiration and fermentation separated in our heads. And for respiration, we have to keep aerobic and anaerobic separate. When we talk about oxidative catabolism of carbohydrate, what we're really talking about is cellular respiration. That term oxidative does not refer to oxygen. It refers to oxidation reduction reactions. And cellular respiration is just one long series of oxidation reduction reactions. It starts with glucose getting oxidized, and it ends with the final electron acceptor getting reduced, whatever that happens to be. In aerobes, the final electron acceptor is oxygen. And in anaerobes, it's some other molecule, typically either nitrate or sulfate or carbonate, although there are other molecules that can also be used. The easiest thing to get confused when you're learning about carbohydrate metabolism is this. Anaerobic cellular respiration is not the same thing as fermentation. You've heard me say it before, you'll hear me say it again. Those are two separate pathways. They involve different enzymes. They both make ATP, but they do it differently. They're both anaerobic. In other words, they don't require oxygen. Anaerobic cellular respiration does not require oxygen. Fermentation does not require oxygen, but they're two completely separate pathways. So a couple other things to remember. All cells, all living cells, microbial cells and other types of cells, are able to oxidize or catabolize glucose. In other words, all living cells are able to break down glucose in order to make ATP. It's just that different pathways are used to do that. Some microbes are also able to break apart other simple sugars, again, like lactose or sucrose or mannitol or maltose. Some microbes can break apart other simple sugars to get the glucose that they will then use to make ATP. That's what we mean when we talk about the diversity of the abilities, the metabolic abilities that these microbes have. Now, it turns out that when we talk about glycolysis, which is at the beginning of cellular respiration and at the beginning of fermentation, when we talk about glycolysis, there are actually some differences in how different kinds of microbes do glycolysis. The idea is still the same. Glycolysis is always glucose becoming pyruvate. It's just that the number of steps and the kinds of enzymes involved in that can differ. I actually put a document up for you on Canvas, a little supplement. Don't worry about it. It's just there if you're interested. But it talks about these different forms of glycolysis that you can see in different microbes. When you look at the MCATs or any of the graduate examinations that students have to take when they want to go on to things like medical school or they want to go to PA school, questions always show up on those exams about glycolysis. So I always provide this little supplementary document to students if you're interested in reading it. It's just glycolysis, but it's happening slightly differently in different microbes. So it gets a different name sometimes. There's just, again, differences between different kinds of microbes. The idea is the same though. Glycolysis begins with glucose and ends with pyruvate. All right. So cellular respiration is a long series of events. It's a long series of chemical reactions. It begins with glycolysis, where glucose will be broken down into pyruvate. Then the second reaction is pyruvate processing. And during this reaction, that pyruvate molecule is going to be broken down to produce a molecule called acetyl-CoA. The acetyl-CoA then goes into the Krebs cycle, and it gets broken down until the final molecule that's left, which is carbon dioxide. So glucose, which goes in to cellular respiration as a 6-carbon molecule, comes out at the end of the Krebs cycle as carbon dioxide, a 1-carbon molecule. All of the bonds in the glucose get broken down, and all of the electrons in there get released. All of those electrons then are going to be used by the electron transport chain to get the energy to make the ATP. Because ultimately, that's what cellular respiration is about. It's about taking apart glucose to make ATP. Now, during the first part of respiration, again, glycolysis, the glucose is converted to pyruvate without needing any oxygen around. So this is the anaerobic part. And during fermentation, this still happens. Glucose becomes pyruvate. It's just that during fermentation, that pyruvate, rather than going into pyruvate processing, like it would in cellular respiration, it's sent down another pathway, a fermentation pathway. And it's converted into some combination of an acid, an alcohol, and or a gas. Now, during cellular respiration, ATP actually gets made 2 different ways. And this gets discussed in the lecture in some detail. A small amount of ATP will be made by a process called substrate level phosphorylation. And that's what this diagram's about. But most of the ATP in cellular respiration gets made through a process called oxidative phosphorylation. Cellular respiration is a great way to make ATP. It's very efficient. For 1 glucose, you get lots and lots of ATP. Substrate level phosphorylation, which again occurs during cellular respiration, it's a very inefficient process. It only makes a small amount of ATP. Fermentation again also produces ATP. That's its purpose. But it uses that less effective, less efficient method. So all of the ATP that gets made during fermentation gets made through the process called substrate level phosphorylation. I also have a note down here that fermentation regenerates a molecule called an electron carrier. Don't worry about that information. The important thing to remember is that the ATP that gets made during fermentation gets made using this less efficient process called substrate level phosphorylation. This diagram walks us through how this happens, how substrate level phosphorylation happens. So this big green blob over here, this represents an enzyme. It can be any of a number of different enzymes. Substrate level phosphorylation can occur at several different times during respiration and during fermentation. So different enzymes are involved. The key thing is that it's an enzyme, so it's going to have an active site where things bind to it. And in this case, right here, the substrate molecule is bound to it. I know it's hard to see, but the artist has drawn the substrate as 3 white squares attached to a yellow circle. So this is some molecule that is the substrate or the reactant in a chemical reaction. It's going to get converted into some kind of product with the help of this enzyme. Now, notice that there's a second molecule attached here, and that's the molecule ADP. During the chemical reaction, whatever chemical reaction this is, because there are lots of reactions where substrate level phosphorylation can occur. During the chemical reaction, the enzyme is going to help convert this substrate into this product over here. You can see that the difference between the substrate and the product is the fact that the little yellow circle is gone. This substrate molecule, the 3 white squares and the yellow circle, this is a phosphorylated molecule. The yellow circle represents a phosphate group. So whatever this is, whatever molecule this is, it has a little phosphate group attached to it. And as the enzyme converts it into a product and then releases the product, what's going to happen is the phosphate group will be taken off of the substrate and put on to the ADP. That's going to turn the ADP into ATP, and that's going to get released as well. There are 2 phosphate groups, 2 yellow circles on ADP. The enzyme's going to take this yellow circle off the substrate and add it on to the ADP. So now there are 3 yellow circles, and now we have ATP, adenosine triphosphate. It's an inefficient process. In other words, each time that enzyme catalyzes that reaction, you get 1 ATP made. Very, very inefficient. It's not a great way to make ATP, but it does make ATP. And that ATP is useful to the organism. Substrate level phosphorylation. So I said earlier that many different sugars can be fermented by different microbes. I've just got a partial list here. The key thing that's getting fermented is glucose, because it's glucose that is being converted to pyruvate. But where the glucose comes from is where fermentation pathways differ. So sometimes glucose comes just as glucose, and the organism takes regular old glucose and ferments it as is. But there are lots of other small sugars, disaccharide sugars, lactose, sucrose, fructose, mannitol, there's lots of them. And some organisms can break apart these small, simple sugars to get some glucose. And then they're going to take the glucose and they're going to send it through glycolysis to get pyruvate, and then they're going to ferment. You need the enzymes. If we say that you can ferment lactose, what we're saying is that you have the enzymes necessary to break apart lactose to get some glucose that you're going to turn into pyruvate, and then you're going to turn into acid, alcohol, and gas. That's what we mean. If you can ferment fructose, what we mean is you can break apart the fructose because you have an enzyme that will break the fructose to get some glucose, which will be converted into pyruvate, which will be converted into acid, alcohol, and gas. That's all we're saying when we say that organisms can ferment different sugars. But again, by learning which sugars they're capable of fermenting, we know some things about that organism. And it can help us identify that organism because not all microbes can ferment all of these sugars. There are lots of different fermentation pathways. And we've learned a lot about them. In other words, we know what enzymes are involved and we know what products get made because we like fermentation, we humans. We have learned how to do it commercially, industrially, to produce products that we like. There are lots and lots of products that get produced through fermentation that have commercial value to us. So we know a lot about all the different pathways. And we push microbes to do them for us so we can harvest whatever product we're interested in selling. You know that yeast can ferment sugars to produce alcohol that we like to drink. We can also get products for foods through fermentation. There are lots and lots of foods that come from fermentation, or food ingredients that come from fermentation, soy sauce and tofu and all kinds of things that we like to eat. There are even things like cosmetic products that come from fermentation. There are solvents that are useful in industry that come from fermentation. So there are factories, if you will, there are commercial businesses that set up huge vats of microbes. They give them all the nutrients they need to ferment. They keep them nice and warm. They keep them in the dark, and they let them ferment. And then they siphon off and purify the product molecules, the acids, the alcohols, and the gases. And they sell them. So we have industrialized fermentation, we humans, by learning about all the different pathways. The two most common fermentation pathways are listed on this slide. In other words, the two fermentation pathways that can be done by the most number of microbes are the lactic acid fermentation pathway and the ethanol fermentation pathway. Remember, you and I can also do lactic acid fermentation in our cells. The lactic acid pathway involves the production of this molecule called lactic acid or lactate. Remember, you're also going to get ATP. The microbe is not doing this to get lactic acid. The microbe is doing this to get the ATP. But we take advantage of the lactic acid pathway, again, in industry. We grow microbes and push them to ferment for us because lactic acid is a product that is used in a lot of food production. Ethanol fermentation is the pathway that produces the ethanol that we like to drink and we like to use in foods. Now, the ethanol fermentation pathway is a little more complex than the lactic acid pathway, but both pathways are pretty simple, at least biochemically speaking. In both pathways, the first thing that happens is glycolysis. The glucose is converted to pyruvate. In all fermentation, that's the first step. Then the pyruvate gets converted into some combination of products, some combination of acid, alcohol, and gas. So in lactic acid fermentation, it's turned into an acid. In ethanol fermentation, the pyruvate is first turned into a molecule called acetaldehyde. So the pyruvate is first converted into acetaldehyde and a little carbon dioxide. Then the acetaldehyde gets converted into ethanol. So an alcohol is the product. In your textbook, there's some good information about all of the different types of fermentation that occur in microbes. I've listed some of the most commonly harvested products of fermentation. In other words, the products that microbes make that humans sell commercially. So, of course, ethanol as the alcohol that we drink, but also alcohols like butanol and one called butane diol. These have industrial applications. There are many other alcohols. This is just a partial list. In terms of the acids, I mentioned lactic acid. Butyric acid can be produced through fermentation. Formic acid, succinic acid, propionic acid. Lots of acids can be produced through fermentation. In terms of the gases, again, there are several, but the two most common that, again, have a commercial value are carbon dioxide and hydrogen gas. On this slide, you're looking at a picture of a table that I took right out of your textbook. It's table 8.3. You can see, again, common fermentation pathways. The name of the pathway is listed in the column over on the left. You certainly don't have to memorize these. I just wanted to give you a chance to see them. The end products of the fermentation pathway are listed in this second column. And as you can see, these are all combinations of acid, alcohol, and gas. Then in the next column, they're showing us some of the microbes that are capable of doing these types of fermentation. And finally, we can see the commercially valuable material that comes from that kind of fermentation. Cheese and vinegar and sauerkraut and yogurt and beer and bread and all those good things. But also things like, again, cosmetic products and solvents that get used in industry. Lots and lots of commercial products can be produced through fermentation. So again, if you're interested, your textbook has more information about these different types of pathways. All right. Does anybody have any questions about anything we've talked about so far? So you should be able to define fermentation. Tell me that it's a process, a metabolic pathway that takes pyruvate that comes from glucose, from catabolizing glucose, and sends that pyruvate down some pathway to produce acid, alcohol, and/or gas. And you should be able to tell me that the reason that microbes ferment is because they need ATP, just like we all need ATP. So I'm going to go ahead and jump back in. This is what we're going to look at today in terms of testing. The first test we're going to look at is simply called the Carbohydrate Fermentation Test. I love tests where the name tells you exactly what they're about. This is a test that's going to help us determine if a microbe can ferment some particular sugar. So this is a test that we can run using all different kinds of simple sugars. We get to decide which sugar we want to test the microbe on. We're going to know if the microbe can ferment the sugar that we put in the tube because an acid is going to get produced. And we're also going to know if the microbe produces a gas. We'll talk about that. The second test has a long name. It's called the Methyl Red Voges-Proskauer test. So, of course, these are the names of scientists that helped develop this test. I hate it when they name tests after people because it doesn't tell you what the test is about. This is actually more commonly referred to as the Mrvp test. And these are two separate tests. In other words, there's a methyl red test and there's a Voges-Proskauer test, but we always run them together. What these tests are going to help us determine is, instead of whether a microbe can ferment a particular sugar, what kind of acid is getting produced during the fermentation process. We're not going to be able to identify the acid by name, but we will be able to identify it by how acidic it is. In other words, this test or these tests can tell apart acids that have a pH less than 4.4, acids that have a pH between 4.4 and 6, and acids that have a pH greater than 6. We'll be able to tell which category the acid falls in. Now, the other thing we're going to know from this is if the microbe can take the acid produced through fermentation and turn it into a molecule called acetoin. Now, you've probably never heard of acetoin. Most people have never heard of acetoin. Acetoin happens to be a molecule that shows up as an intermediate in a lot of metabolic processes. So whether or not a microbe can produce acetoin is just one of their characteristics. And the Mrvp test is going to tell us whether or not they're able to do that, whether they can take the acid that is produced during fermentation and further metabolize it to produce acetoin. Now, the last test we'll look at is called the Citrate Test. This is a nice simple one. In this test, we're going to examine whether or not a microbe can ferment a molecule called citrate. If it can ferment the citrate, we're going to see a color change in the media. Now, what makes the citrate test really interesting is that citrate is not a carbohydrate. Citrate, or citric acid, is another one of these molecules that shows up in a lot of metabolic pathways. For example, it shows up in the Krebs cycle. It's part of the intermediate molecules that get produced during the Krebs cycle. It's not a carbohydrate. So we always think about fermentation being all about carbohydrates. But it turns out that some microbes can take that citrate and turn it into pyruvate, and then send the pyruvate through the rest of the pathway. So that's an unusual trait, if you think about it. And we're going to test to see whether or not microbes have the ability to do that by feeding them some citrate. All right, here's the list of the organisms we're going to use today. You can see our old friend E. coli on the list. Remember, E. coli is a gram-negative rod. It lives in the intestine, so we refer to it as an intestinal or a enteric or a fecal organism. It's also considered a coliform. You'll see that term a lot. All a coliform is an intestinal type of organism, or again, fecal organism, that can ferment lactose. The second one is called Enterobacter aeruginans, also gram-negative, also a rod, also lives in the intestine, also a coliform. So you can see these two organisms are pretty closely related. Third is an organism called Klebsiella Pneumoniae, also gram-negative, also a rod, also a fecal coliform. We're sensing a pattern here. Klebsiella is an interesting organism because Klebsiella is, in some cases, pathogenic. There are some pathogenic strains of Klebsiella pneumoniae, just like there can be pathogenic strains of E. coli and pathogenic strains of Enterobacter. The difference is that Klebsiella pneumoniae, when it's pathogenic, it doesn't only cause disease in the intestine. In fact, it's well known as causing pneumonia, hence the name, meningitis, and also cellulitis, an inflammatory infection in the skin. So when it's pathogenic, it's a bad bacterium to have to deal with. It causes some very serious diseases. Now, next on the list is an organism called Shigella sonii. That's how you say that species name. Shigella sonii, you will sometimes see referred to as Salmonella sonii. These are the same organism. It's just that it has undergone a name change. In fact, it's undergone a couple of name changes in my professional life. Scientists are always, trying to reclassify things. Drives the rest of us crazy. But Salmonella and Shigella are so closely related genetically that this particular organism keeps going back and forth. People say, no, it's a Salmonella. No, it's a Shigella. So this organism has gone through a name change a couple of times, back and forth, which is why, depending on the source materials that you read, sometimes it's referred to as Shigella sonii, and sometimes it's referred to as Salmonella sonii. It just depends on what source material you're using. So we're going to call it Shigella because I think that's the most recent name that it goes by. This organism, this Shigella, is also a gram-negative rod, also fecal, but it's not a coliform. So what that means is it cannot ferment lactose. I think the most interesting thing about Shigella is that it can get into your cells. So it doesn't just cause disease in your intestine, living in the cavity of your intestine. This is a bacterium that can get into your cells, into the cells that line your intestine. So when you get a pathogenic strain of Shigella sonii, it can cause some pretty serious disease. Now, last on the list is Pseudomonas, Pseudomonas aeruginosa. This is a gram-negative rod, again, fecal or intestinal, but not a coliform. And Pseudomonas aeruginosa is an opportunistic pathogen. This is a type of pathogen when you get a pathogenic strain of it that doesn't normally cause problems for healthy, active, fit people. It becomes a problem in people who are compromised, people who are already sick, people who are in the hospital because they've had a heart attack, or they're in the hospital because they've been in a terrible traumatic accident, or they've been burned over some significant portion of their body. So they're compromised. Their health has been compromised. Their immune system is very busy. It's taking care of these other problems in the body. And Pseudomonas takes advantage of that. That's what opportunistic pathogens do. They take advantage of the opportunity to cause disease. So Pseudomonas can be a real problem in chronically ill patients or patients who are acutely ill with a serious disease. It's a terrible organism. When people get infected with it, it's a terrible organism. It can be hard to treat. Okay, so we've got 3 gram-negative coliforms, and we have 2 gram-negative non-coliforms. All of them live in the intestine. All of them, at least in certain strains, can cause disease. Now, the first test we'll look at is this carbohydrate fermentation test. And what you're looking at on this slide is a commercially available form of this test. So you can buy these tubes that come with the carbohydrate fermentation media already in them. You can order whatever sugar you want them to put in. You can order glucose in here. That's the simplest test. Or you can ask them to put in any particular sugar you want to test. You can also make your own carbohydrate fermentation medium. You can mix it up in the lab. But a lot of people buy this medium already made. They buy it from a commercial source because the other thing that this test has involved in it is this device called a Durum Tube. So you can't really see it because the medium is red, but there's a little glass test tube in here called a Durum Tube, a little tiny one, and it's upside down inside this bigger test tube. The Durum Tube is used to see if any gas gets produced while the carbohydrate is metabolized. So here's an empty test tube that has a Durum Tube in it. Can you see it? It's right here. It's just a little glass tube, but you put it in here upside down inside the test media. And when you grow the microbe in here, as it ferments, if it releases any gases, some of the gases are going to get trapped in here. The gases, you remember, are going to rise up to the top, and some of them are going to get trapped in this little Durum Tube. So again, you can make your own carbohydrate fermentation medium, but a lot of places buy it commercially. Now, here's how you interpret this test. Remember, just like any test medium, you're going to take the tube that you start with and you're going to inoculate it using your very best aseptic technique. You're going to use a sterile loop to gather up some cells. You're going to open up this tube while you're holding it at an angle. You're going to take your loop and you're going to put the cells down in here and wiggle it around a little bit. Take your loop back out. And then you're going to close the cap again, put it in the incubator and let it incubate. Now, when you take these tubes out the next day, what you're looking for is color change. So over here on the right, this is a tube that is a negative result. In this tube, the medium has not changed color, right? It's still that red color. And this is actually a control tube. So no cells were put into this tube, but the tube was put into the incubator. In other words, it was used as a control, an uninoculated experimental control. Remember, we make controls because we want something to compare the other tubes to. So these 2 tubes were inoculated with something, some kind of organism. And as you can see, there's been a color change. It's turned to yellow. There's an indicator dye in here called phenol red. And under acidic conditions, it turns yellow. Now we also have to look at the Durum tube, and I know it's hard to see, but here's the Durum tube sticking out of the test media. You can see that there's liquid all the way up to the top of this Durum tube. So whatever's growing in here, it's a positive test for acid production. That's what the A stands for. It's a positive test for acid because the medium turned yellow. But it's a negative test for gas because there's no gas in that tube. This one is different. This is a positive reaction for acid, tube is yellow, and it's positive for gas production because look, there's a little gas bubble up at the top of the Durum tube. See that? So whatever's growing in here was not only able to ferment the sugar to produce an acid, but it also produced a gas. So we always run an uninoculated tube. And by run, I mean we inoculate our test tubes, our experimental tubes, and we put them in the incubator. And we're also going to put in a tube that has not been inoculated. And that one's going to be our control. We want to put our control tube through everything that the experimental tubes go through. So if we're going to put the experimental tubes in the incubator, we want to put our control tube in the incubator too. And we're going to use our control as our comparison tube. Controls are really important. Sometimes, especially when you're doing a test that uses an indicator dye, so you're looking for a color change, sometimes these tests go wrong. Sometimes, media gets old and indicator dyes get old. So controls are important, not only as a comparison, but also just to make sure that your experiment went well. In other words, if I took a control tube, an uninoculated tube of carbohydrate fermentation medium, and I put it in the incubator along with my other tubes, and I came back the next day and I took all the tubes out, if that uninoculated tube has turned yellow, I've got a problem, don't I? Something's wrong. I didn't put any cells in that tube. It shouldn't have changed color. If it did change color, that tells me I need to throw all those tubes out. Something went wrong. Something went wrong. It could be that my experiment got contaminated somehow. It could be that the indicator dye has gone bad. But if my control doesn't do what I expect it to do, which is nothing, then I know I have a problem. So controls are very important. They're a very important part of an experimental process. All right. So we know what a positive result looks for acid, and what a positive result looks for acid and gas. Now let's take a look at some of our organisms growing in a carbohydrate fermentation tube. Now this just happens to have glucose in it as the carbohydrate. Now I put this picture in because I want to show you what it looks like when you make your own carbohydrate fermentation medium. Remember, this is what it looks like when you buy it commercially. The phenol red gives it this nice red color before the tubes are inoculated. This is some homemade carbohydrate fermentation medium. And this is a control tube. So this is the uninoculated tube, what the tube looks like, what the medium looks like before you put cells in it. As you can see, this is not red. This is orange. And here's why. Part of the reason that we made the carbohydrate medium from scratch is because we wanted to add an extra ingredient in. We wanted to add peptone in for this particular experiment. And when we add peptone in, it changed the color of the medium. Again, it's really important that you have an uninoculated tube for comparison. because it didn't start red. It started orange. So this tube over here, which happens to have Pseudomonas in it, this is a negative result. Why is it negative? Because there's no color change. There's no difference between the control tube and the Pseudomonas tube. So sometimes when we opt to make our own media in the laboratory, it's because we want to mix it up special. We want to make some change to the normal recipe. And anytime you alter the recipe of a medium that has an indicator dye in it, you're going to change the starting color a little bit. But that's okay, because the way you interpret the test is to look for a color change. And we can certainly see that even with this homemade medium. You can certainly see that a couple of these tubes have changed color. This one here has E. coli growing in it, and this one has Shigella sonnei growing in it. We've got a color change. We've got a change to yellow, which is indicating that there's acid in here. And if you look very carefully, you can see that there's also gas in the E. coli tube, in the Durum Tube. So E. coli has not only fermented the glucose, but it has also produced a gas. The Shigella sonnei has fermented the glucose, but it did not produce any gas in that process. And the Pseudomonas aeruginosa is negative. Now, for the next several slides, we're going to look at some results for that list of organisms I showed you a minute ago. I don't actually have pictures for you. They did not come out very good. So instead, I have some diagrams of the results. Okay, so this is a comparison of carbohydrate fermentation test using glucose, lactose, and sucrose. So 3 different tests, 3 different tubes. Each tube has a different sugar in it. And we're going to compare E. coli and Klebsiella here. Here's the control tube. Here's what the media looked like before it was inoculated. All right, so we had 1 control tube for glucose, we had 1 for lactose, and we had 1 for sucrose. And then we took media and we inoculated it with E. coli. We inoculated a glucose tube with E. coli, and a lactose tube with E. coli, and a sucrose tube with E. coli. And then we did the same thing with the Klebsiella. Klebsiella went into the glucose tube, Klebsiella went into a lactose tube, and Klebsiella went into a sucrose tube. The reason I'm combining these 2 organisms on this slide is because they both had identical results. Here's the glucose result for both organisms. Here's the lactose result, and here's the sucrose result. Yellow, 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 so acid. And bubble, 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 so gas. All 3 of these are A/G. Positive for acid production, positive for gas production. So both of these organisms, and you can see this at the very bottom of the slide, both of these organisms, E. coli and Klebsiella pneumoniae, can ferment glucose, they can ferment lactose, and they can ferment sucrose. And for all 3 of those sugars, they produce both an acid and a gas. Very adaptive little organisms, very adaptive little bacteria, because they can ferment and produce ATP from a range of different sugars. They may be able to ferment other sugars as well that we didn't test. But we know that both organisms can ferment these 3 sugars, which is, an interesting trait of those bacteria. This is what Shigella Sonnei did on that same range of tests. So we tested Shigella Sonnei in glucose, in lactose, and in sucrose again. And here's the results we got for that organism. The glucose tube, we got a positive for acid and a positive for gas, just like we saw for the other two organisms. But we got no reaction in the lactose tube and no reaction in the sucrose tube. So no color change, and so obviously no gas. So Shigella is not able to ferment lactose, and Shigella is not able to ferment sucrose. And that's what I've written down here in that little table. Acid and gas for glucose, no reaction for lactose, no reaction for sucrose, not able to ferment those sugars. Here's the Pseudomonas. Looks pretty similar to the Shigella, doesn't it? We got a positive result for the glucose, negative for lactose, negative for sucrose. But there's an important difference in the glucose tube. Remember, Shigella was able to ferment the glucose and produce both an acid, that's the yellow color, and a gas. Pseudomonas can ferment glucose, but it does not produce a gas. In other words, Pseudomonas is using a different pathway here. It's fermenting glucose to produce an acid and no gas. It's just a different pathway than what the Shigella used. They can both ferment glucose, but they do it differently. So I put all those results together for us right here. E. coli, Klebsiella Pneumoniae, these are two coliforms. They're two coliforms, so we know that they can ferment lactose. But we also now know they can ferment glucose, they can ferment sucrose. Shigella Sonnei, see, even I do it. Shigella Sonnei, Pseudomonas aeruginosa, no, they're not coliforms. They cannot ferment lactose. They are also not able to ferment sucrose. They don't have the enzymes necessary to take the lactose and break it apart into glucose and galactose. They don't have the enzymes necessary to take the sucrose and break it apart into glucose and fructose. So yes, they can ferment glucose, but they can't get glucose out of these other sugars. And Shigella produces a gas during fermentation, while Pseudomonas does not. All right. Questions about the carbohydrate fermentation test. We typically buy this medium, but we don't have to. We can make it. When you buy it, you just tell the manufacturer what sugar you want in it. If you only want to know whether or not the organism can ferment, just put glucose in it, because it's the glucose really that's being fermented, right? It's the glucose that's going to be used to make pyruvate and then whatever product molecules get made. But you can also ask the manufacturer to put other simple sugars in there if you want to test the microbe's ability to ferment several sugars. You can see I've got a uninoculated carbohydrate fermentation test tube here. Again, commercially sourced. You can see the Durum tube in there, especially if I play around with it a little bit. The Durum tube is in there, but it's full of fluid. It's full of the medium, so it's not that easy to see. Now, one of the things that is important to remember about this, when you buy these commercially, is that sometimes they get jostled around in shipment. And what'll happen during all that jostling is that some air will get into the Durum tube, Can you see the air bubble? It's like half the length of the Durum tube. If air gets into the Durum tube, you've got to get the air out before you inoculate, because this air was not made by a microbe. This air just was in the tube and got trapped because the tube got jostled around during shipping. So what you have to do is kind of roll the tube back and forth, and you got to encourage the gas bubble to come out. It's important before you inoculate this test tube that there's no gas in the Durum tube, because I want to know if my microbe can make gas. So I don't want to start off with any gas in there. Okay. And then this is the color we're looking for a positive result for acid. And this also has gas in the Durum tube. See that? So you inoculate them, you put them in the incubator. Next day, what's the color? Is there a change? Compare it to your control. And is there any gas in the tube? And then you know whether or not that microbe can ferment that particular sugar in order to get acid, alcohol, and or gas. All right. So we're going to switch gears here and look at this next test. Remember, this one is called Mrvp. It's actually two tests. It's an MR test and a VP test. It's a Methyl Red Test and a Voges Proskauer test. So I'm going to have two different tubes. Now, the other unusual thing about this is that we're introducing a new indicator dye. We're not using phenol red here. We're using methyl red. We're still going to be looking for a color change, but it's going to be the reverse of what we're used to. So that's why it's important to remember. Methyl red test uses methyl red as the indicator dye. And methyl red is going to start out yellow and turn red. So that's exactly the opposite of what we've seen, right? Normally we expect the medium to start red and turn yellow if there's any acid, but this is exactly the opposite. This medium begins yellow. So we're going to look for a red color to develop as a positive result. Now, what the methyl red test is doing for us is telling us not if an organism can produce an acid through fermentation, but what the pH of that acid is. So we're going to look for the yellow starting color to become a nice cherry red, like this one, if the acid that is being produced through fermentation is a very acidic one. It has a low pH, a pH less than 4.4. If the acid that's being produced, the product acid, has a pH less than 4.4, you're going to get a cherry red color change. If the acid that gets produced has a slightly less intense pH, if it falls between 4.4 and 6, what you're going to see is a more orange color. And then you can also see an acidic positive reaction that is an orangey yellow color if the product acid has a pH of 6 or more. So this test especially, you have to have control tubes to use as a comparison, because you gotta be able to tell if the color is different from the starting color. And if it is different, is it cherry red? Is it red orange? Or is it orange yellow? All this test is going to tell us is the pH of the acid product, essentially. Now, the VP test is different. The VP test, we're not just going to see a color change. This is what VP medium looks like before you inoculate it. It's a pretty awful color, isn't it? It's kind of a beigey green color before you put the cells in. Now, you're going to put your cells in, and then you're going to put the tube in the incubator. When you come back the next day and you take the tube out of the incubator, it's going to look exactly the same. This medium already starts off cloudy. So even after the cells grow in it, it's still going to be cloudy. You're really not going to see much of a change the next day. Now, what you have to do is you have to add in a reagent chemical. You have to put in a few drops of a reagent chemical in the tube after you take it out of the incubator. If the top of the tube, where that liquid hits, if it turns red, you've got a positive test. If it doesn't turn red, you've got a negative test. And remember what the positive test means. It means that this microbe growing in here has fermented the sugar and produced an acid, and then converted the acid to acetone. So the reason we run these together is because they're really asking related questions, aren't they? We typically are asking, we typically already know that we've got an organism that can ferment. Maybe we did the carbohydrate fermentation test first. So now we want to know, well, what's the pH of the acid product? And can it take the acid product and turn it into acetone? So we run these together because the two questions we're asking are related to each other. Now, you can see from the list of organisms that we started with today and the ones that we ran through the carbohydrate fermentation test, there's one organism missing from our list. There were 5 that we said we were going to work with today: E. coli, Klebsiella Pneumoniae, and Enterobacter aeruginans, as well as Shigella and Pseudomonas. So I didn't do the carbohydrate fermentation test for Enterobacter. I saved that organism for the Mrvp test. Both E. coli and Enterobacter aeruginans are coliforms. They're both gram-negative rods, intestinal organisms, both coliforms. They're closely related to each other. They can be hard to tell apart. But one of the tests we can use to tell them apart is the Mrvp test. So what you're looking at on this slide in these pictures, these two are the result tubes for Enterobacter and E. coli for the Methyl Red Test. So these are the Methyl Red tubes. And over here on the right, these are the VP tubes. Again, Enterobacter, E. coli. So let's think about what we're looking at. Remember, the MR test, the test medium starts off yellow. I don't have a control tube in this picture, but this tube, the A tube that has the Enterobacter growing in it, this tube did not change color. So what does that tell us? That tells us that the Enterobacter aeruginans did not produce an acid, did not ferment the sugar and produce an acid that we could evaluate on this test. Negative results for Enterobacter aeruginans on the MR test. E. coli, on the other hand, produced a positive MR test. Nice cherry red color. E. coli grew in here and fermented the sugar and made an acid with a pH less than 4.4. So big difference here for these two organisms that are closely related. Negative for Enterobacter, positive for E. coli. If you look at the VP results, it's exactly the opposite. Enterobacter gave a positive result. E. coli gave a negative result. Enterobacter is positive on VP. E. coli is negative. So if you put the results into a table, you can really see why the Mrvp test is useful for distinguishing these two organisms. It's also useful for many other organisms, but it's especially useful to tell these two apart. E. coli is going to be positive on MR, negative on VP. Enterobacter is going to be negative on MR, positive on VP. All right. Questions? The important thing to remember about that VP test is that when you take the tube out of the incubator, you have to add a reagent in. There's a little, like a squeeze bottle that comes with the test. And you have to put 2 or 3 drops in to the tube when you take it out of the incubator. If there's any acetone in there, the top of the tube is going to develop a red ring. That's the most important thing to remember about that test. You'll pull those tubes out in the morning and they're all still that beige green color. But then when you add the reagent, you see, for some, you see a red ring develop. All right, the last test that we're going to look at today is the citrate test. Now, this particular auger that we use for this test, it's technically called Simmons citrate auger. So again, this was named after the person who invented it or first described it. So again, citrate testing or Simmons citrate auger, this is typically done in a slant. Remember that this diagonal surface of the auger we're going to streak some cells right down there with our inoculating loop. This media starts off with a very sort of deep, dull green color. You're going to spread your cells down the surface of the slant. You're going to inoculate the tube, in other words, put it into the incubator, take it out the next day, and look for color change. And in this case, we're going to look for this brilliant blue color to appear. So after I inoculate the tube, if the tube stays green, I've got a negative. This organism cannot ferment citrate. If the tube turns blue, this organism can ferment citrate. Now you can make or purchase Simmons citrate medium as a broth, but most people that I've encountered anyway, most people use it as a slant. It's just super easy. On this slide, instead of putting the Simmons citrate auger into a slant, what we've done is we poured it into a Petri dish to make a plate. Now, the reason we did this is because I wanted to put both organisms on the same plate. So this is what the uninoculated Simmons citrate auger looks like. Again, sort of a dull green color. And all I did was I drew a line down the center to make 2 sides to the plate. I streaked Enterobacter aeruginans on the left, and I streaked E. coli on the right. Again, these 2 organisms that are both coliforms and both, closely related to each other. And once again, we have a difference. You could see the Enterobacter side turned that brilliant blue color. Enterobacter aeruginans can ferment citrate, but E. coli can't. So the E. coli side stayed green. Just to be complete, I will show you what Simmons citrate looks like as a broth. You can see this is just a broth. This is just a liquid. But this is a positive test. So it is possible, instead of using a slant, to just use a broth instead. Just inoculate the broth with cells, leave it overnight, and look for the color change. This is an uninoculated slant tube of Simmons citrate. Again, dull green. I don't have a positive slant tube to show you, but I do have the positive broth tube to show you. Really brilliant blue, really very different from the original starting color. So this organism was able to ferment the citrate. And the indicator dye lets me know that because of the very significant color change in the tube. All right, so the last bit of results here, again, are just to show the difference between these two closely related organisms. The E. coli had a negative result on citrate. It cannot ferment citrate, but Enterobacter aeruginans can. So both of these tests, the Mrvp and the citrate, are very helpful for distinguishing these two organisms in the laboratory. All right. Any questions? Again, citrate's not a sugar. It's not a carbohydrate, so you don't usually think about things fermenting non-carbohydrates. But it is possible for some microbes to ferment citrate. Remember what fermentation means. It means taking pyruvate and turning it into acid, alcohol, or gas. So some microbes can use citrate to make pyruvate. Most of the time, though, we're thinking about an organism using glucose to get to pyruvate. And then the pyruvate is converted into a product. All right. So very, very common types of tests that we run in the lab: carbohydrate fermentation tests. The actual one, called the carbohydrate fermentation test, allows us to, really customize the media, and we can test whatever sugar we want to test to see what kinds of traits our microbe has, so that we can identify the microbe. Because what sugars the microbe is able to ferment is a trait. It's a characteristic of the microbe. Mrvp is a little different. Mrvp, we're asking more like, what kind of acid product is being produced in terms of its pH? And can the microbe turn that acid into acetone? Mrvp. And then finally, citrate, we're asking, can this microbe use citrate to ferment, to make pyruvate, and then to make either acid or alcohol and or gas? All right. Any questions at all about this? All right, good job. Good job, everybody. If any questions come up as you're looking at the homework assignment, the lab questions, just send me a message. Remember, the lecture material this week it's difficult. It's difficult material. I recognize that. So make sure you give yourself plenty of time to watch it so you can take a good set of notes. And if you have any questions while you're going through it, write them down, send me a message, okay? And we'll talk about them. All right, that's all I have for you today. If there are no questions, I'm going to let you go to enjoy your evening. And I will see you again next week. All
Thank
right,
you.
everybody. Have a
Thank
good night.
you.
You're welcome.